Sure. I am a resident in general surgery at the University of Pennsylvania with an interest in thoracic surgery. And so that's cancers of the chest, primarily the lungs and the mediastinum as well. Well, I think it can be challenging, especially because these nodules, lung cancers present early on as very small nodules in the chest. And so sometimes, even with our advanced screening and imaging modalities, they can be challenging to identify. And then even if we see something on a CAT scan that's concerning for lung cancer, it can be challenging to actually biopsy those lesions. And so I think the combination of these being very small lesions that could be challenging to see both on CAT scans and then also actually biopsy to determine if it's a cancer or not is the primary reason why why early stage lung cancer can be a challenge to detect. Absolutely. So we conducted a early stage feasibility trial of a new technology aimed at being able to detect lung cancer in very small quantities. And so this was primarily done in cell and animal models of lung cancer. And what the technology is it uses a targeted tracer that actually glows, and so it will localize to cancer cells because cancer cells overexpress certain cell surface receptors, and so this kind of glowing molecule localizes the cancer cells, and we give that to the animals before the biopsy procedure, and then during the biopsy we actually use a small kind of detector that can actually pick up this glowing signal from the cancer cells. And this detector is so small that it can fit within the middle of the needle that we use to actually take the biopsies. So essentially, we're able to, using this technology, to detect cellular level quantities of cancer during biopsy. And so the goal is basically to be able to use this technology as a way of assisting traditional ways of biopsying cancer in order to increase the accuracy in being able to detect cancer earlier. So as I said, we primarily did this in cell and animal models of lung cancer, but we did test this on human tumors after they'd been taken out of the body, and we're able to show that it works in human tumors as well. So we're excited for the possibilities of this work. Ultimately, we need to do this in real patients with their tumors kind of inside of their lungs still, to be able to determine if definitively whether it works or not. But we're excited about the possibilities. I think we were excited by the ease of use of this technology. I think a lot of times some of the technologies that we study in the lab are challenging to implement in a clinical setting because they're not very user-friendly. But with this technology, you can actually you can take your biopsy, you can stick this device in the biopsy needle, and you can either see basically one of two things. Either you can see glowing cancer cells, or you can see essentially a blank screen. And so it's very easy for surgeons and for radiologists or pulmonologists who are taking these biopsies to actually determine whether or not they have the cancer or not in the biopsy. And so we're excited that this may actually have real clinical relevance and may be able to transition this technology into clinical use at a more rapid pace than previous things we've developed in the lab that are a little bit more limited from a practical application standpoint. Well, I think the ultimate goal of the work of our lab generally is to be able to improve outcomes for patients with lung cancer. And the best way we know how to do that is to be able to detect lung cancer earlier. And so there are a lot of efforts that have been going on nationally in order to detect lung cancer at an earlier stage, primarily by having patients undergo CAT scans if they're at high risk for lung cancer. And so in that way, we're able to pick up a lot of lung cancers earlier. But then once you pick up these nodules that are suspicious for lung cancer, in order to actually determine whether or not it's a cancer or not, you need to biopsy it. And so that's kind of really where we come in, is to be able to take these millions of nodules that are detected annually and be able to more accurately tease out which ones are cancers or not, and which ones can undergo curative surgery. And so I think the goal of our efforts, in combination with a lot of efforts that are going on around the country, is to improve the detection rates and early treatment for patients with lung cancer in order to have an impact on their survival in the long term.